Hi class, let's talk about fungi, mushrooms, and all that jazz. There are lots of fungi, and this is a chapter 4 discussion, so we're talking about eukaryotic cells. All of our fungi are eukaryotic cells. And there's two big categories of fungi. The ones you can see with your naked eye, the macroscopic fungi, and the ones you can't see, the microscopic fungi. When you think of a traditional mushroom, a puffball, or a steppe fungi, sometimes also referred to as a gill fungi, those are the macroscopic fungi. Those are the ones that you, generally speaking, are going to chop up and put in your food, so to speak. By the way, puffballs, they taste kind of weird. I'm not a big fan of puffballs. Anyways, our microscopic fungi, molds or yeasts, as their name implies, are really small. You can't see them with the naked eye. We still add quite a bit of yeast to our foods. Molds in smaller quantities, particularly if you're a big fan of blue cheese, you will like having mold in your adding mold to your food, but generally speaking, we try to shy away from the mold. There are over 50,000 species of fungi that have been identified. This number is continually growing larger. Once we shifted towards genomic identification of fungi, we have been finding new species up the wazoo. I read an article just last fall about a gentleman who went to an oriental market in Chinatown and bought some mushrooms off the shelf and just ran them through his thermocycler to synthesize the DNA, and he discovered a new species of mushroom that had never been identified before that was being sold at the supermarket. So this number of 50,000 species is just going to skyrocket now that we've been using genomics to isolate species. Our fungi come in different forms. They can be single cellular or unicellular. They can be in a colony that can exist by them, where cells can exist and live by themselves, but prefer to live in a group. And then finally, we can have those macroscopic fungi, those large ones that we can see with our naked eye. They're complex, they're multicellular. Those are the ones that you traditionally think of as a mushroom or a puffball. So let's talk about yeast. I want to emphasize this. Yeast is a fungi. Yeast is not a bacteria. When somebody has a yeast infection, when somebody ferments beer, when somebody makes yeast bread, they are using fungi, not bacteria. Why am I emphasizing that so much? Because it took me an embarrassingly long time in my life to figure out that fungi, or that yeast was not a bacteria. So hopefully, now you know, yeast is a fungi. Yeast are typically going to be single cellular or unicellular organisms that have an ovoid or round shape. They typically will go through asexual reproduction through a process known as budding, which is similar but distinct from binary fission, which bacteria use for reproduction. The other category of cells are going to be the hyphae. And instead of being these cells that can stand alone, hyphae are going to be long thread-like cells that will form the main body, the main trunk, or the volume of our filamentous fungi. If we have yeast cells growing in a chain, or if we take some unicellular fungi or spherical fungi and line them up in a row, they can look like they're a filament, but they're really individual discrete cells, and that's called a pseudohyphae. To tell the difference between a regular hyphae and a pseudohyphae, you're going to need a light microscope with very high resolution. Generally speaking, most introductory light microscopes for most undergraduate institutions don't have the precision that you need to differentiate a hyphae from a pseudohyphae. But at least you know the definition of it. Some fungal cells will be dimorphic. When I say dimorphic, you see this word di meaning to, morph meaning shape, so they are two-shaped cells. They can take either form. They can be either a single cellular cell or they can exist in a hyphal form, depending on their growth conditions. So here we see not a hyphae, but we can see some individual yeast cells. And these individual yeast cells are kind of spherical in nature, and we can see some budding occur. So this one cell all by itself is having little baby cells pop off of it, grow off of it. And then the baby cell can start to have another baby cell grow off of it. If you're a Star Trek fan, you might have watched the classic episode, The Trouble with Tribbles. The trouble with tribbles is that all tribbles are born pregnant, and they start reproducing even before they're done being fully grown. And this is what we see happening here with our fungal cells. Our fungal cells will immediately start the process of budding a new cell before the individual cell itself is done growing. Moving on. There are not as many fungal species that cause disease in human beings, thank goodness. So we have over 50,000 identified or known species of fungi. Only 300-ish of those species are capable of causing disease in human beings. So what's that in terms of a percentage? That means that less than 1% of known species of fungi is capable of causing disease. So most fungi don't cause disease, thankfully. And this is assuming that you don't eat them. If you eat them and they're poisonous, that doesn't count. There are three kinds or categories of fungal diseases in human beings. We have the community diseases. These are ones that we can acquire from a social gathering. Think of like locker room environments or dirty wrestling mats or gymnastic mats. Those are going to be the community acquired infections. Ringworm is a good example. We could have environmental pathogens that are present in the general population, or we could have hospital associated or nosocomial fungal infections. Harmless spores or harmless fungal spores that normally for a healthy individual won't cause infection, for someone that is immunocompromised can cause a full-blown infection. For example, if somebody is an AIDS patient, acquired immunodeficiency syndrome patient, they could have fungal infections that a normal person with a normal functioning or a person with a fully functioning immune system wouldn't necessarily express. This is to the point that fungal infections have been used as diagnostic infections for AIDS patients. We'll talk more about that later when we have our section on AIDS, which is in the second half of the course. I don't want to get ahead of myself here. Now, if we look at the fungal cell wall, just to emphasize what we talked about earlier, is made of a complex polysaccharide known as chitin, and it's also capable of giving off substances that cause allergies. This is that time of year where those of you that are allergic to black mold just go, oh man, I need to get out of the basement. And then you go out of the basement and there's pollen everywhere and you go, oh, I just can't escape. I'm allergic wherever I go. So yes, those substances given off by fungi are absolutely capable of causing allergies. Some mushrooms that are toxic are capable of inducing death. For example, if you eat the death cap, that could be bad for you. A black mold infection or fungus known as Aspergillus flavus is going to make a poison that's potentially lethal to animals. This is a mold that grows on grain products. So for those of you who work in agriculture or have a background in agriculture, you probably remember going through the silage or going through the feed and going, oh man, this stuff has black mold on it. I need to throw it away instead of feeding it to my cows. Aspergillus flavus is the fungal species responsible for you throwing away that moldy grain because you don't want to have sick cattle. Not a good thing. If we look at fungi in agriculture, many species of fungi don't have any effect on us as human beings, but instead are going to affect crop species. Corn smut is an excellent example of this. Corn smut is unique in that it's a fungi that infects a plant, an agricultural species that we, or I shouldn't say we, but some cultures consider a delicacy. If you go to the Hispanic section at larger grocery stores, you can actually find canned corn smut. They don't call it corn smut. I forget the Mexican name for it, but it's considered a delicacy and, as an aside, tastes kind of leathery to me. So some fungi can contaminate corn or other grain products, and if our livestock eat moldy corn or grain, they could get sick. No big surprise there. What is alarming, though, is how much of our produce, we as human beings, the human produce that we lose due fungi from shipping. We lose approximately 40% or nearly half of all the food that we put in a box or we try to ship is destroyed or rotted by fungi during the transit process. That's a big problem. I know for any of you that store winter squash in their basement, and I'm one of them, you typically lose about 40% of your squash due to fungal rot. I know this winter I tried putting a bleach solution on my squash to see if they would last longer, and they didn't. We lost a ton of squash this winter. Fungi aren't all bad. They can do some good things, though. They help us decompose organic matter. If it wasn't for fungi, we would be covered by meters upon meters of undecomposed wood on our planet. But because we have fungi that are constantly decomposing dead wood, they will help recycle that organic matter through the carbon cycle. And some species of fungi are going to help us by forming associations with a symbiotic relationship with plant roots. Plant roots that have the correct fungal associations are able to absorb and retain more water and absorb and retain more nutrients. A classic example is the legumes for crop species. Legumes are a species of plant that are known for having nitrogenous fungi in root nodules, and is one reason why many farmers will alternate between having a legume and then a non-legume crop year after year, every other year on their field, so that those fungi can help fix nitrogen into the field. Fungi can be genetically engineered. It's very easy to genetically engineer, particularly a strain of yeast, to produce a specific molecule, your favorite molecule. And that's just by taking the specific gene in question, throwing it into a plasmid, throwing that plasmid into the fungus, and then reproducing it like crazy, and then purifying out the extract. Some things that we've engineered fungi to make for us include antibiotics, alcohol, organic acids, and vitamins. Some fungi we can eat directly. Think of your portobello mushrooms, your morel mushrooms, or sometimes they're going to be used to impart flavoring to food. For example, think of the mold in a blue cheese. Now, fungi, being living organisms, need to have some way to get nutrition into their body, some nutrient source for their body. Nearly every single fungi is heterotrophic. When I say heterotrophic, I mean that they can eat a wide variety of organic substances. They are not autotrophic. They do not make their own food, but heterotrophs need to eat their food. And so if we look at heterotroph here is a big umbrella term. Underneath the umbrella of heterotroph, we have saprobes and we have parasites. A saprobe is a fungi that obtains its nutrients by eating dead organic matter. So a saprobe is a heterotroph that eats dead organic matter. A parasite is a heterotroph that eats living organic matter. If we look at a fungus in general, the fungus in general, as it's consuming its organic matter, is going to do most of the digestion extracellularly. So instead of phagocytizing the big chunk of dead wood and then digesting the chunk of dead wood inside of the cell, the fungus is going to secrete digestive enzymes outside of the cell. Those digestive enzymes break down the food, and then the fungus will phagocytize small packets of organic matter at a little bit at a time and bring it into the cell. Because fungi are capable of digesting their surrounding environments and absorbing the surrounding environments, these fungi can traditionally survive in very nutritionally poor or harsh environments. Fungi are going to be associated with many pioneer species. If you look at the series of succession, if you start out with a bare rock, typically the first thing that's going to start growing on a bare rock is a lichen. And a lichen is a symbiotic relationship between a fungus and an algal species. These fungi can grow on the bare rock, or they can grow in a high saline or a high sugar environment. Typically, when your jams and jellies go bad, it's because of a fungal infection or a fungal invasion. We spent a little bit of time talking about the morphology of fungi when we had their individual yeast cells, or we could have the hyphae. Most fungi are going to grow in a unicellular state, or a very loose association of colony of cells that can break off and live as unicellular cells. If we look at a yeast colony, it's almost identical to a bacterial colony from a macroscopic perspective. What do I mean by macroscopic perspective? I mean, if you look at a chunk of yeast cells growing and a chunk of bacterial cells, and this glob is big enough for you to see with the naked eye, it's going to look nearly identical, squishy and slimy. Sometimes your fungal species are going to have, instead of having a colony of yeast fungi, you'll have a colony of filamentous fungi. And if you have lots and lots of little tiny hairs or filaments, as you could imagine, the macroscopic view is going to be kind of hairy or velvety looking. So let's talk some more morphology or fungal anatomy. If we look at a mycelium, the mycelium of a fungi, and here's the mycelium right here. The mycelium is the part that you cannot see. It grows beneath the surface. The mycelium of the fungi is going to make up the main body or mass of the fungi. This mycelium, generally speaking, is going to be interwoven with the soil or interwoven with the host, and is not relatively available to view. The mycelium should be differentiated with the vegetative hyphae. The vegetative hyphae are going to be that mass of hyphae that you can see with the naked eye. So when you think of a mushroom, think of a vegetative hyphae. So this fruiting body up here is made of vegetative hyphae. Now, in between individual cells within the mycelium, within the hyphae of a fungi, we're going to have little cross sections separating adjacent cells. These cross sections that separate adjacent cells within a hyphae are called septae. Now, these septae are unique in that they allow organelles and nutrients to flow from cell to cell. So they aren't a very good barrier. They can differentiate or separate cells from each other, but they still allow adjacent cells to have communication through their cytoplasm. For example, here's a hyphae, and this hyphae has lots of individual septae separating the individual cells, distinguishing individual cells from each other. But we can still have organelles and cytoplasm transfer between adjacent cells. Some hyphae are going to be non-septate hyphae, and if they don't have that septae, they're going to resemble one ginormously long cell. They will be a multinucleate cell. A good analogy with this would be a skeletal muscle cell. And then finally, the reproductive or fertile hyphae are the specific hyphae that produce the mold spores. So if we were to look at this figure right here that I pulled from the internet, the reproducing or fruiting hyphae would be the gills on the underside of the mushroom's cap. Hyphae can reproduce and form spores through a variety of methods. All mushrooms can reproduce via hyphae. So if you chop up a mushroom and take the little bits of hyphae from the mushroom and spread them out all over the place, that will propagate the the fungus. However, we can also have fungal reproduction through spore generation. You can think of a spore as the fungal equivalent of a gamete or a sperm and egg. Our spores are going to be responsible for reproduction, particularly sexual reproduction for the fungi. Our spores can be dispersed through a wide variety of environments. So they can be airborne, they can be waterborne, or they can be transmitted through living things. These spores can germinate upon finding a favorable substrate, or when they have ideal or favorable environmental conditions, and they can start a new fungal colony in a relatively short period of time. If we have an asexual spore formation, we are going to start out with a structure known as a sporangiospore. A sporangiospore is made by having the hyphal cell cleave multiple times. And as it's cleaving multiple times, it's going to cause a sac known as a sporangiosperm to form. The sporangium. The sporangium is the ball at the end of the stick, or the ball at the top of the tower, so to speak. And the sporangium is what gives off the spores. The sporangiophore is the tower that holds it at the top. And if you take a sporangium and combine it with a sporangiopore, you call it a sporangiospore. Yeah, talk about a tongue twister. I know. So, here we have the sporangium. The sporangium is the sporangiophore, and the sporangiospore at the top. Sometimes, instead of having sporangiospores, we'll have conidospores or conidia, depending if you're singular or plural. These are free spores that don't need to be enclosed by that spore-bearing sac. They don't need to have the sporangium enclosing them. So over here, we can see our conidospores. So instead of having that sac holding them all in, they will be allowed to free float and enter the environment. We can also have sexual spore formation. Sexual spore formation is an ideal way to form spores for a fungi because it will have more mixing and matching of genes to give us potentially favorable trait generation in the population. So we'll have genes from two parent fungi that could be linked together. As these genes are being linked together, they'll create new offspring with new combinations of genes. This is a good thing. Sexual reproduction is good for the populations. This is why most major animals and most major organisms that we can see, most multicellular organisms, go through sexual reproduction. These variations can lead to advantageous adaptations, so on and so forth. Sexual spores can vary from a simple fusion of two parent cells combining together to a complex union of male and female parts combining with each other. So depending on the species of fungus, we could have simple sex or complicated sex. So, concept check class. From which of these sources can fungi derive nutrients? Can fungi derive nutrients from dead plants and animals, living tissues, rubber, petroleum products, or E? All of the choices above are correct. You should check your textbook for this one. You can also scroll back in the presentation or rewind this video. This wasn't covered very explicitly in this lecture presentation. It is covered in more detail in your text. So pause it and go look up the answer. Okay, hopefully you've looked up the answer. The answer is E. All of the above choices are correct. That's all we have for this presentation. Happy studies!